Good afternoon, participants of the 5th webinar from the HealthFerm project. We are now going into the details of omics technologies in food fermentation processes. We have a very short introduction of Professor Christophe Courtin. Then we give the floor to Professor Stefan Wex. After him, we have the floor for Kati Hanhineva, and then Dr. Azadeh Mohammadi will give the perspectives of the industrial part. These are the titles of the presentations. Christophe Courtin will give a very short introduction on the HealthFerm project so that you know the context of the presentations. Professor Wex will give a presentation on how genomics can improve food fermentation processes. Professor Hanhineva will go into the details of the metabolomics of food fermentation processes, and as already said, Azadeh Mohammadi will provide some insights in shaping the future of the bakery, participatory and chocolate industry through omics technologies. I have some household messages. That is, the presentations will follow without interruption. If you have questions, there is a possibility to address questions through the system, the question and answer box. We will follow those questions carefully. Please address the question to the presenter you want to ask your question to. And at the end of the webinar, we will collect the questions and will address the questions to the presenters. And we will try to address the most urgent questions, and if your question is not answered, then we will answer it by following up by email. The webinar is recorded, so you will also be able to watch it back and see what is of your interest. These are the household messages, and I would like to give Christophe Courtin the floor to give his short introduction on the project. Christophe, please go ahead.
Thank you, Jan. And I'll just try to find the right screen. And I hope it is this one. Can you see my screen?
Yeah, your screen is on, Christophe.
And you only see my screen, right? Nothing else hidden?
I see your screen and I see your camera.
Which is not a good thing, or?
Oh no, that's what I want. Yes, please.
Okay, perfect. So welcome everybody to this webinar on omics technology, and I'm very happy to provide you with a very short introduction to the HealthFerm project. You might have seen this before because I do this for every webinar. Now, very shortly, so HealthFerm is a project that consists of 23 partners from 10 countries. Is a project that lasts for 4 years and is funded by the European Union and also by the Swiss SERI. And the names of the different partners I'm not going to read through. You can see them on the lower left-hand corner of your screen. But I think you recognize a lot of the different logos of a lot of different good partners that we have in the project. Now, what is HealthFerm about? HealthFerm aims to enable an evidence-based transition in society and industry from traditional to sustainable plant-based fermented foods and diets. So what we want to do is to help the transition from animal-based products to plant-based products and to make the transition to a more sustainable food and diet system. We want to do that evidence-based. And very important is that we want to look at health benefits. Yeah, so we want to make sure that we want to try to produce food products that have health benefits by design. Now, for this, we need microbial resources, we need fermentation technology, we need knowledge on the interaction between human health and the human microbiome and food microbiomes, and we also need knowledge on consumer behavior. And if you put that in a graph or in a figure, this is what you get, so the different elements. And what you might know or not know is that in the last 2 years, we have given different webinars as HealthFerm, and they have focused on either microbial resources, on fermentation technology or the interaction, looking, for example, at sourdough, looking at different microorganisms, how they interact with specific substrates. We have also focused on work package 3, so giving a webinar on human health and how fermented foods can interact with human health and also with the human microbiome. You might remember that we also did a piece on consumer behavior, and we will do one again later this year. But for today, the most important thing is that we're going to look at omics technologies. So we have over the 3 different work packages, 1, 2, and 3, a lot of omics technologies involved, be it metabolomics, be it genomics, and others, to help us understand what is happening in those in different processes within different microorganisms or in the microbial community in our gut. And so that's the topic of the webinar today, and I wish you a lot of pleasure listening to the different speakers that we have lined up for you today. So
thank you very much. Thank you very much, Christophe, for this very nice and short introduction. I would like to invite Professor Stefan Wex to come to the floor now. Let's call it that way. Stefan, floor is yours to provide your introduction on omics technologies.
Okay, thank you, Jan, and good afternoon, everyone. In my part of the seminar, I will focus on the use of genomics to better understand microbial strains, which then can help us to make rational choices of starter cultures. Yeah.
We don't see your screen. Yeah. Now we see your screen. Okay, this is go.
Okay. There is some delay then. So to make rational choices of starter cultures for food fermentation processes. So I will first briefly introduce some concepts on genome sequencing and annotation, and followed by some concepts about comparative genomics. I also will discuss a case on comparative genomics that is sort of related. Then I will zoom into genome mining and to wrap up with some conclusions. Okay, so if we look at the definition, what genomics is, genomics is a study of the complete set of genetic information in an organism, so the genome. So how do we obtain a genome of an organism? That's by DNA sequencing. So here at the right-hand side of this slide, we see a timeline on the x-axis, and on the y-axis, the number of sequence entries in public databases. And you might have heard already of the several generations of DNA sequencing. So the traditional Sanger sequencing is considered the first generation. And then since 2004, 2005, there is an increase in data also because the introduction of first, second generation sequencing methodologies, so short read sequencing, and then later on what we call third generation sequencing, so long read single molecule sequencing. When we then look specifically to genomes, we see kind of the same trend here in the green line, a steady increase of complete chromosomes of organisms. And then somewhere 2004, 2005, a steeper increase of contigs and scaffolds. So that means there is DNA sequence available for organisms, but not to the complete status of the genome. When we look nowadays, what is currently available with a high throughput, that's third generation sequencing with, on the one hand, Oxford Nanopore Technologies. We see here pictures of their Promethean platform. So with such a chip, you can sequence 100 to 200 gigabases of native genomic DNA, and their kits allow to combine barcoded 96 samples. Base accuracy of ONT is nowadays 99.75. The competitor, as to say, is PacBio. So with their Revio, you might not see it here from this slide, but the ONT platform, it's a benchtop platform. the Revio is really a large machine that just stands on the floor. They have their high fidelity reads, 120 to 180 gigabases per smart cell, and their barcoding system allows to combine up to 384 genomes in one run. Bacterial genomes is the number, and the base accuracy is slightly higher. It is 99.95%. Okay, so once you have sequenced the DNA of an organism of interest, it comes to the next level, and that's the genome assembly. So reconstructing the original genome, so chromosome or a replicon. So you have the reads, they assemble into contigs. Contigs can assemble into scaffolds, which means that it's a kind of ordered set of contigs, and then at the end, you can reconstruct the genome. Either it does not succeed very well, so you end up with maybe a very large contig and very small contigs, and maybe even a plasmid. And then sometimes you are able to assemble into a circular chromosome with maybe some leftovers, or just one nice big circular chromosome for bacteria. Next step is to find the genes and to annotate the genes. So I've here depicted a flow chart for the PROCA pipeline, which combines, in fact, gene prediction. So not only the coding sequences, but also transport RNA, ribosomal RNA. And then Prodigal is used specifically for the protein encoding genes. And then the genes in the genome are then aligned to databases, for example, Swissprot, to also perform protein annotation, so to know the functions of the genes that are present in the genome. The output of PROCA is typically text-based files, so either GenBank files or some more specific output files for this kind of pipelines. That is not very human readable, so you can use it on servers and to process with scripts. If you want to really have a view on a genome, then I recommend you to have a look at Ensemble Bacteria. So that's one of the genome viewers available on the internet. We look here at a part of Limosilactobacillus fermentum IMDO130101, a strain I will give you some more details later on. So it's slightly above 2 gigabase pairs, and this is just a section of the genome. We sequence a lot of genomes altogether. So luckily there is an institute, NCBI in the US, that keeps track of all those genomes in their genome database. At this moment, for the family Lactobacilliaceae, there are slightly less than 16,000 genomes available in this database. It's either complete or at scaffold or contig status. Searching in this bunch of genomes, so they're not all food related or fermented food related, is only a small number, a couple of hundreds that are food fermentation related. Of course, when searching for that kind of keywords, you have to rely on the annotation and the metadata that the colleagues have added to that genome. Okay, so that brings me to comparative genomics, because there are a lot of genomes available in those databases. So if you have a species of interest, you can do comparative genomics, for example, for taking into account several strains of that same species. So conceptual wise, when we do comparative genomics, there's some terminologies like we speak about the core genome. And we see here a very nice graph depicting 3 organisms. And we see here that the core is a common set of genes that is present in all 3 organisms. Then the dispensable genome, it's a set of genes that is shared by a few, in this case, 2 by 2. If it's a more complex comparative genomics, you have much more combinations. And then the unique genome, it's the set of genes that is unique for every one of those organisms. The full collection of genes that is present in all these 3 organisms, that's what we call the pan genome. Now, a couple of years ago, we did some genome sequencing of strains collected from cocoa fermentations. And these are even 3 different species, so Acetobacter ghanensis, pastorianus, and senegalensis. And what we knew from fermentation experiments is that pastorianus always prevailed, and that ghanensis and senegalensis, after a while, were not growing out any longer. So we see that I have a core genome of 1,733 genes. And then, of course, if you know that, for example, this pastorianus strain prevails, is much more competitive than the others, it was, of course, of interest to have a look at these 700 genes. And we found indeed genes and pathways that gave this strain a competitive advantage, and it had to do with resistance to low pH conditions in cocoa fermentation. Okay, so that's one way of doing comparative genomics, looking at the genes. You can also have a look at the genome itself, and that's what we see here in this slide. So here at the left-hand side, it is what we call a synteny plot for 2 genomes. So at the x-axis, it's the genome of Acetobacter pastorianus. At the y-axis, it is the genome of Acetobacter ghanensis. And every dot here in this graph corresponds to a specific gene that is present on a specific location in the genome of Acetobacter pastorianus, and then also at a specific location in the genome of Acetobacter ghanensis. The y-axis is a kind of percentage of the length of the pastorianus genome. What is very interesting here, that although these are 2 different species, we see that there are always a stretch of genes that are in both genomes in the same order. And that's, of course, very interesting also when you want to look at metabolic pathways. Another level of looking at comparative genomics is to have a look at DNA polymorphisms. So here at the right-hand side, we see part of 2 genomes, where we see very nicely a translocation of a genomic region, and also in red and in blue, insertion deletions, so small parts of the genome that is present in one of the genomes but not present in the other genome. So a few years ago, Marco Verse, one of our previous PG students, did a comparative genomics analysis on the Lactobacillus fermentum IMDO 13101. So that's an isolate from a spontaneous laboratory rice sourdough fermentation. So it had a good growth in that sourdough. It ferments maltose, produces mannitol, and is rather good acid tolerant. So it is one of our candidate sourdough starter cultures. So we did a comparative genomics with 27 other strains that were available at that time in the genome database. So 8 were plant-based, 5 were milk-based, and we see here the colors in this table. So the green, it's the plant-based. The yellow ones are what we call milk-based, then 12 human-related isolates, and then 3 isolates with an unknown source in white. So when doing a phylogenomic analysis, so we see here at the left-hand side a phylogenomic tree with the same color codes as in the table of the previous slide. So this phylogenomics tree was based on analysis taking into account 584 single copy core genes. So these are genes that only once in the genome, but present in all of these strains. And we could reveal that all these cluster in 5 different clades. When we take the genome sequences and perform an average nucleotide identity analysis, also the 5 clades appeared also here in that ANI analysis. When we look here at the colors, we can conclude that there was no clear clustering based on isolation source. So that suggested that Lactobacillus fermentum had a free living lifestyle. That the largest clades contained most of the human and milk related strains, and that our strain here, 130101, was in a clade together with 3 other plant-based strains. So Marco also made a representation of metabolic path, so the carbohydrate metabolism. And remember, our strain was in clade 2, so it had indeed some very specific features that most likely were the basis of that competitive advantage in sourdough fermentation. So here we see the proof of the genetic potential to metabolize xylose, arabinose, beta-glucans, and alpha-glucosides. And it also can oxidize here pyruvate into acetyl phosphate. So compared genomics is in fact looking at the full genomes, comparing all genes and reconstructing eventually metabolic pathways. When you're interested in some specific gene or a set of genes, then you can go towards genome mining. And I'm very happy that I can share this already. It's a paper from Ines Pradal in our group that was accepted about 2 weeks ago. So on the production of esters by lactic acid bacteria. So esters provide fruity notes to fermented foods, among which sourdough. But the ester production by LAB under the conditions that are similar to those that occurring during sourdough production, it's still questionable. So our research question was like, is ester biosynthesis by LAB possible from the free precursors? So the background knowledge that is already available was that there were 3 esterases, EST-A, EST-B, and EST-C, that have been studied under comparable conditions as sourdough. EST-A was already proven to be in Lactobacillus helveticus, and then EST-B and EST-C were proven to be in lactic-casei Bacillus casei. Also, the biosynthesis of the ethyl esters was shown in vitro, so ethyl butanoate, ethyl pentanoate, and ethyl hexanoate, and also in a cheese model. So there was already evidence to start from. So what Ines did is she looked into 401 genomes of lactic acid bacteria. So she checked the genome database and searched for all strains of species that were described in sourdough. So it didn't mean that all those strains were indeed coming from sourdough, but at least the species was ever already described to be present in sourdough. So she used the amino acid sequences from the EST-A from Lactobacillus helveticus and the amino acid sequences of EST-B and EST-C of lactic-casei Bacillus casei to perform a T-BLAST-N alignment versus a translated nucleotide database. So this figure is in fact one figure representing all that work, and it's quite a dense figure. So we see here in the middle the phylogenetic tree of those 401 genomes. But I want to point your attention to these outer circles, and especially the most outer circle, which represents in black those strains that harbored the EST-C gene based on similarity search. And I hope you also agree with me that the outer circle has relatively more black parts than the others. So it looks from this graph that EST-C is most present in these, of all the 3 genes, that EST-C is the mostly present one. So indeed, and if we would look further into detail, the 3 genes were present and the presence was species dependent. And company Lactobacillus paralimentarius and Crustorum and Limosilactobacillus fermentum were sourdough relevant species containing at least one of those genes. Now, since this genome mining was based on sequence alignment, we also went back to the gene annotation to check like, okay, what is the annotation of the gene in those specific genomes? And then we had to conclude that the annotation was rather generic, stating alpha, beta hydrolases. And that's also what we see here in this graph. So EST-B and EST-C were annotated as alpha or beta hydrolases. Now, I just mentioned these species names that had at least one of the genes. Interesting was also that Fructilactobacillus sanfranciscensis, like one of the, let's say, famous sourdough species, did not harbor any of those esterase coding genes. So next step was to look in the culture collection here in the research group. So Ines sequenced the genomes of 12 strains of our collection, had to look at the presence of those 3 genes. And indeed, also in this very small subset of strains, we already saw that the SC gene was present in all these strains that we have here in-house. So of course, proof of the pudding is in eating. So Ines also did in vitro fermentations to assess the ester biosynthesis by those selected LAB strains. So she used a modified wheat sourdough simulation medium with ethanol with acetic acid with a combination of several organic acids and several higher alcohols. So these are the graphs. So the left graph is the ethyl acetate concentration and the right-hand graph is ethyl lactate concentration. So here we see that Company Lactobacillus castorem produced most of the ethyl acetate, followed by several strains of Company Lactobacillus paralimentarius. And the ethyl lactate was mostly produced by strains of Company Lactobacillus paralimentarius. So also here, no ester biosynthesis by Fructilactobacillus sanfranciscensis strain, so that actually confirmed what we already saw with the genome mining, so pure in silico analysis. And we assume that SC was involved indeed in the ester biosynthesis. So to answer our research question, yes, specific lactic acid bacteria can synthesize esters, be it that their ester biosynthesis capabilities is limited by the precursor concentrations. For more details, I kindly refer to the publication. So to conclude, I hope I have convinced you that next generation sequencing combined with bioinformatics analysis allows us to unravel genome sequences of individual microorganisms to better understand their functional role in food fermentation processes. That comparative genomics provides us insights into the traits related to the functional role and also the competitiveness of specific strains in an ecosystem. And that genomics information enables us to make more rational choices regarding the functional starter cultures to produce plant-based fermented foods. So to finish, I would like to thank our team members, my colleagues, especially Koen Illegems, Marco Verzi, who are previous PhD students, and then 2 current PhD students, Gabriela Samaniego and Ines Pradal. And of course, I would like to thank all our partners and funding organizations, and I thank you for your attention.
Thank you very much, Stefan, for an impressive, though short presentation. I see that there are not that many questions at this moment, so it's either very clear or it raises so many questions that it's difficult to answer. However, let's see what our next speaker, Kati Haneniva, has to present on metabolomics of food fermentation processes. Kati, floor is yours.
Thank you so much. Please let me know when you see it. Yeah, it's in the presentation.
Okay, thank you.
Thank you. I will still organize my screen a bit. Okay, so good afternoon, everyone. It's a great pleasure to be joining the HealthFerm webinar today and tell you about the metabolomics technique. So I will first give a short introduction to metabolomics in general, and then I have 2 already published works how we have utilized metabolomics in assessing the fermentation process of various food products. You see the slides changing, right? I assume so.
Yeah, slides are
Yeah,
changing.
okay, very good. Thank you so much. Okay, so metabolomics, as the name tells, is focusing on the analysis of metabolites. So we heard just about genes. So if you take all the genetic material, DNA and RNA out of your sample and also extract proteins, then whatever is left in the sample you can address with metabolomics techniques. So it's the general profiling of all the metabolites that can fall within the analytical technique that you are using. And in food science and nutrition in general, it is typically used to study the biochemical composition of various different foods and the impact of processing on it. And also, on the other hand, we are interested in what kind of impact on our inborn metabolism and relevance to the gut microbiota, for example, these metabolites have. And for that purpose, we can also utilize metabolomics to profile, for example, gut-derived samples or blood samples and so forth. And metabolomics analysis utilizes technologies such as mass spectrometry and NMR. And these techniques have been around for several decades, of course. And you might ask that, what is actually the new point here? What is the novelty, or what is different in metabolomics when thinking about the normal traditional targeted quantitative analysis with LCMS, for example? So the difference is that when we do the metabolomics analysis with, for example, LCMS, we are trying to collect all the signals that the instrument can show us. So we don't have any particular compound. We are not looking at any named amino acid that we want to measure, but we let the instrument show us whatever there can be detected. And for this reason, the sample preparation is typically very generic, only precipitation of the genetic material and proteins, and then without any further processing there. So we try to keep as much compounds in the sample as possible. And typically metabolomics analysis, when you do it this way, in a non-targeted profiling way, they are semi-quantitative. So you don't get exact quantities for each of these, but you do see the relative ratios. So you can compare that before fermentation, after fermentation, how large difference each of the different metabolites have there. And therefore, it is typically without any targeted pre-hypothesis. The general hypothesis typically is that my treatment has an impact on the metabolite profile, or this dietary intervention will affect the plasma metabolite composition. So that is a general. And the greatest advantage of this is naturally that when you don't draw the exact hypothesis in beforehand, you leave the door open for such findings that you wouldn't see if you have only the targeted compounds in measurement. And in our research group, we have utilized, first of all, let's go through briefly this workflow still very briefly. So as mentioned, any biological material can be utilized. Typically in our case, that is many times food samples, or then some samples derived from animal feeding trials, internal organs. It can be fecal material or plasma samples or whatever. And the sample preparation is very straightforward. And then after that, we inject it in mass spectrometry analysis. And in our case, we utilize 4 different analytical approaches. This is for the purpose that we want to cover as wide metabolite coverage as possible, because different metabolites have very different chemical properties. So if we use reverse phase and hydrophilic interaction liquid chromatographies together, then we have much wider polarity range that we can address the metabolites. And in addition, we also ionize the metabolites with positive and negative ionization, so that will cover, increase the coverage of the metabolite signals that we can detect. Then after the actual instrumental phase, which is typically can be quite rapidly accomplished, then comes the data processing side. And this first we need to pre-process the data. We need to take all the different samples that have been analyzed in the instrument. We need to collect the data, transfer that to numeric form, and then put everything to one data matrix. And for this purpose, we utilize different, it can be a software that is freely available, or it can be a vendor specific software. And currently we are using mostly the MS-Dial open source software. And we combine this with a package called Notame, which is a pre-processing and quality control package that has been developed in our laboratory. And once the data matrix is ready and cleaned up and the quality has been ensured, scaling, transformation, whatever different kind of procedures are required there, then we can conduct statistical evaluation on the measured signals and start finding such metabolite signals that are showing differences between the different treatments. And only then we will then start identifying what these signals actually are. And this last part, identification, that is typically where we spend majority of the time in a metabolomics data analysis procedure, because a lot of the signals, especially coming from such rich matrices as food products, many of those are such that they are not yet listed in any databases, so it can be sometimes relatively tedious process to actually then give a name to these components. And here is a scheme of the general scheme, what kind of different approaches we have utilized the metabolomics analysis in our research group. And we cover quite widely the whole pipeline, from all the way from the raw materials to the very end when the food is processed and entered human body and finally excreted. And this is just to demonstrate that how we can see that how different the metabolites can be when they start from the plant when it grows, and you can have a lot of different effect on their composition by the cultivation conditions. And naturally then the food technological processes can have very various biological reactions, especially such as fermentation, where we use microbial material that transforms the metabolites. We can have a lot of differences occurring on the metabolites. And then once the compounds enter our body, then all the way from the mastification and upper part of the GI tract, and then especially in the colonic microbiota fermentation, various different conversions can occur to the metabolites. And then finally, when they are excreted, the metabolites when they enter the body, they can be in relatively different form. That's what they originally were in the plant. And metabolomics approach is good to give the overall view and monitor and follow these different routes all the way in this pipeline. And then let's go to the examples from our earlier studies. So actually both of these studies, the key author is Ville Koistinen. So this one comes from his PhD work. This was published already 2018. And here we conducted metabolite profiling analysis on sourdough fermented wheat and rye bread. Here is the study setting for this study. So we had two different wheats as a starter material, both for the wheat, two different flours for the wheat and two different flours for the rye. So we had whole grain rye and whole grain wheat, and then both of those were refined. in refined form, wheat and white wheat and shifted rye, as it is called. And then doughs were processed, and from the whole grain flours, we also produced the sourdough fermentation in addition to the straight dough. Baked the breads, and then actually in this the whole study setting, we also then entered all these different breads to the in vitro human digestion model. But now we will focus on what kind of differences we had in the sourdough baked bread and samples related to there. So typically when we conduct a metabolomics analysis, the first general view on the data is principal component analysis. Here, this scheme, this represents now all the different metabolite signals that have been collected, something like I would say around 5,000 metabolite signals most likely behind this scheme. And the first difference that is obvious here is that wheat samples and rye samples are clustering quite distinct from each other, so each dot is one sample. And the flour samples are distinct, both the refined and whole grain flour, both wheat and rye. And then when the baking starts, it can be either the sourdough or the straight dough baking, there is a huge shift then on the metabolite composition. And especially then in the case for sourdough rye, we can see that the whole grain sourdough, the dough itself, and then the baked bread, they were very clearly differential from the rest of the analyzed samples. And if we take a little bit closer look on what kind of metabolites were altered in this sourdough baking process. So here in this scheme, we see the metabolite levels in a bubble plot, how they were behaving in the sourdough rye fermentation, and the x-axis here represents the retention time in the chromatography, and y-axis is then the fold change in the sourdough. And up here, these are the signals which were not detected at all in the yeast fermented rye, but only emerged after sourdough. And here you see a large pool of different components that are having differential levels in the rye sourdough, and many of these are written in a blue, marked in blue color, and those are mostly amino acids and peptides and so forth, and some phenolic components as well. And some of those phytochemical metabolites were such that we already knew would be, or we expected to be emerging. So for example, the derivatives of the phenolic acids, ferulic acid and synapic acid, were in a reduced form as a dihydroferulic acid and dihydrosynapic acid. So there was a clear shift in the metabolite composition in the rye sourdough baking. But then when we take the same look on wheat sourdough, we were a little bit surprised to see that actually the metabolic differences, the conversions, they were clearly less pronounced as they were in the case of the rye. So a couple of metabolites were such that they were occurring simply, or they were only detected after the sourdough fermentation, and some of those were similar, these derivatives of the phenolic acids, but not as metabolite dense as it was in the case of rye. And then in general, the overall look, how many metabolites, how many different components we could observe. There were upper part here depicts the significantly increased molecular features, and then the lower part are those that were decreased in the process. And when comparing the whole grain rye sourdough and whole grain wheat sourdough, you could see that both the increased and decreased metabolites, they were always much more detected in the rye than in the wheat, and only a couple of tens of metabolites were such that they were overlapping between the different fermentations with the different grains. And here is then a heat map view, and it's very difficult to read, I know, because it's very dense in the different components. Here at the lower level of the screen, I hope you can read, this is the legends for the different samples, different sample types. And for example, this area, you can see that these are metabolites that are always formed when yeast fermentation is conducted here, so somehow specific for yeast fermentation. And then when we move to the right hand side of these plots, so this is like a continuation, so it's the same order of the samples. So these two columns here, they are depicting the rye sourdough samples, the bread and the dough. And as you can see, here is a huge amount of different signals that come only in the sourdough rye. And majority of these were different amino acids and different D or tripeptides that were released from the matrix. So it clearly demonstrated that there is a massive turnover occurring during the fermentation of both the wheat and the rye, but especially in the case of the rye, obviously it is not only the phytochemical metabolites that are converted, but these peptides most likely come from the breakdown of the proteins in the present in the rye whole grain rye flour. And what our conclusions on this general view on this first publication was that it's very clear that the sourdough fermentation causes massive turnover on the biochemical pool, and this one could call as postbiotic signature. So as this terminology nowadays includes this postbiotics, which are the metabolites or the biochemicals coming after the fermentation processes. And these signals, they do remain there even after the baking process, even when the product no longer contains any live bacteria. So probably you have also been involved in discussions where there are sort of doubts about whether sourdough bread should be even included in this fermented food category, or because it doesn't contain the prebiotic bacteria live anymore. But of course, these products, they still contain the massive metabolite load that the fermenting microbes have contributed there. So absolutely it has relevance. And the one reason for having this difference between the whole grain rye and whole grain wheat might be that since in this study we followed the typically applied conditions for sourdough fermentation, and whole grain rye typically has a little bit higher temperature and longer time in the fermentation than wheat. So this might be the reason why then in the case of whole grain rye also the fermentation of the proteins started to occur, and the accumulation of amino acids and peptides was much more evident for the rye than in case of wheat. And of course, it's very interesting to speculate that the metabolite pool coming from the sourdough fermentation could actually have some relevance also then from the health beneficial effect side. And especially we have been focusing a lot on the lowered postprandial insulin response after eating sourdough rye than with whole grain wheat, and this is one of the approaches where we have various clinical studies also ongoing and investigations ongoing, so that could there actually be some trick in the sourdough fermentation that would have then these health beneficial effects in terms of the insulin metabolism. And then if we move to the other example that is also conducted by Ville, and this is one of the studies he has performed during his postdoctoral studies. And this one was conducted in collaboration with our Swedish collaborators, so Maria Hedberg and Anders Johansson from Umeå University, and then the team of Rickard Lamberg from Chalmers Technical University. So this is now focusing on a particular microbial strain, Lactobacillus plantarum, that is a very commonly used strain in rye fermentation. And here we took a multi-step analysis on this particular strain. So the background here is that it has been observed that this particular L. plantarum strain, as you can see here, our colleagues in Umeå, they had different studies where they showed, as in this microscopic figure can be seen, that this particular strain has a very strong adherence to the rye bran, and it always accumulates in the rye, and it has an excellent growth there and seems to be thriving when in presence of rye. And also they could show that this same strain it can inhibit the adhesion of Helicobacter pylori, so harmful bacteria occurring in the gut. And for this reason, they were then interested to investigate further what kind of metabolic processes are related here and what could this compound do. And then in this setting with our collaborators in Sweden, we could then perform a multi-step study where we conducted then for the metabolomics side. And we set 3 different study questions in this study. First of all, we want to know what kind of metabolite pattern is produced by this particular strain. What can it actually metabolize from the rye bran? And whether we can see the same metabolites in any of those real-life sourdough rye products, bread that we eat. And if we can even see similar metabolites also present or occurring in our colon. So we had also an in vitro colonic model included here. And then whether these metabolites that we see produced, can they have any impact on inhibiting the growth of harmful bacteria? And here, very briefly, if we go through the study setup, so we had first the plantarum alone. We took the samples and did metabolomics analysis. Then we had a sourdough fermentation time series, collected samples there. Then we had different sourdough and also yeast fermented rye breads. We collected samples there, and then we had on an agar plates the inhibition tests, and then finally also checked the in vitro situation. And when looking at the metabolite composition, we could see that there were many different metabolites that were produced in the incubation with the L. plantarum alone. And also we could see that there were many different metabolite conversions in these different rye-based sourdough breads. And among these, we could see that there are 7 different metabolites that were always present there, both in the fermentation of the plantarum and also after the sourdough fermentation. And these 7 metabolites represented very typical or well-predicted metabolites that can come from the microbial conversion of compounds present in the rye bran. Derivatives from hydroxycinnamic acids, derivatives from aromatic amino acids, and derivatives from branched chain amino acids. And these 7 metabolites we took into closer examination. We took a look on the in vitro model. Do we see any of these there? And here, these graphs you can read show that the line plots, they are always from the in vitro model. So you can see the fecal control in the bottom, and then the whole grain sourdough rye bread added in the in vitro model with the red line. And then the bar plots demonstrate the different sourdough breads. And you can see that these same metabolites, they are in the dough, in the bread, and then they also increase in the in vitro conditions, demonstrating that these are really such metabolites that are plenty in our food and in our system. Then these compounds were tested against different bacterial strains on the agar plates. The plantarum strain was as a positive control, and as you can see with the yellow color, there was no inhibition when adding these compounds on the plates. But then all the other bacteria, when we added these different metabolites to the plates, they had somewhat inhibition. And then when we added all the metabolites, all the 7 metabolites as a mixture, there was a clear inhibition on various different harmful bacteria that were cultivated on the plates. And adding ferulic acid and caffeic acid, the source metabolites there, it didn't impact this effect too much. So it was already obvious when these microbial converted metabolites were added on this system. So it was very clearly demonstrated that these same metabolites that we can have in the sourdough and in the in vitro system, they can inhibit on the plates these growth of different harmful bacteria.
Kati, can I ask you to finalize in a few minutes?
I am there, yeah.
Oh, thank you.
Yep. So to conclude this second study, we could see that similar metabolic conversions were occurring during the food fermentation and colonic fermentation, and that these metabolites that were produced by the L. plantarum, they had a synergistic effect when compared to the gut bacteria on the harmful bacteria, so that they could diminish their growth. And naturally, there are also other components that can be there, and also we don't know yet whether these are specific actions to the rye, but can this be then a situation for other fermented foods as well. And here I would like to acknowledge the research groups in University of Turku and University of Eastern Finland, on also our collaborators, and also then thank the different funding agencies that are behind the work of our group. And thank you very much for your interest.
Thank you very much, Kati, for a very interesting lecture on metabolomics. Very impressive what you were able to do in your laboratory. I'm looking forward to the questions that people will ask, because I think there are many questions with respect to the work that you're doing. But let's first see how we can use the methodologies as well in the food industry. Azadeh, please go ahead.
Thank you. Do you see my screen completely?
I see your screen, but we don't see the presentation yet. Yeah, now we see presentation. Yeah. Great, thank you.
I'm just, yeah. So good afternoon, everyone. It's a pleasure to present you today, and I hope by the end of this presentation, you will feel inspired on how we use omics technologies to tackle some of the challenges we face in our industry and how we drive innovation. Before we start into the science, so basically at Paratus, we offer a full range of solutions for the sector of bakery, patisserie, and chocolate. By solution, I mean ingredients and both services. So everything that you see on this slide, it's a potential Paratus product. We are present worldwide through 85 subsidiaries and more than 50 distributors, and the headquarters is based in Belgium, where I'm speaking to you today. So as mentioned, we have a worldwide presence with 119 innovation centers, which are places where we present our solutions to our customer, where we receive them and eventually co-create solutions with them. The company has a net sale of 3.1 billion euros. These numbers are from 2023, and we invest more than 2% of our revenues in research. So as you see in our annual sustainability report, we care about sustainability, and there are 6 pillars. One of them starts with health, which mentions the importance of the health dimension that we associate to our product and our way of working. The second one is heritage, to which I will make references during my presentation. And it's not clearly mentioned in this wheel, but fermentation plays a key role in the category that we call heritage, health and well-being, but also to reduce CO2 emissions by the replacement of animal-based solutions toward plant-based solutions. So one of the main challenges that we care about is how we can feed 8.5 billion by 2030 with nutritious and sustainable food. And the way we do that is by leveraging the power of fermented food. So on one hand, we have the advantage related to food preservation, leveling power, especially for bread that are brought by fermented food, and especially the taste and flavor that is boosted in fermented food. On the other hand, we have the nutritional aspects, the nutritional value, and the positive impact on digestibility and gut microbiome. So, as you know, omics techniques are a whole set of technologies which allow to dig deeper into the molecular components of a sample. On one hand, we have genomics and microbiomics, which allow us to identify who was or is there. So basically strains, species, and genes that was nicely presented by Professor Wex at the beginning of this webinar. And on the other hand, we have a whole set of techniques which focuses more on specific molecules at proteins, such as proteins, enzymes, lipid metabolites, which was highlighted, part of it was highlighted by Professor Hanhineva, Katina in this presentation. So basically, with this right combination, we are able to find out who is there and what they do. And this is basically what will guide us into addressing the different challenges that we face in the industry. So as you see on this slide, basically we can use this the omics techniques to address the nutritional improvement, the functionalities, new functionalities, to explore the root cause analysis of food safety and qualities, to trace and find the authenticity of the raw materials, but also the traceability of the fermented food, and also to boost the flavor and sensory analysis. So in the first part, I will focus on the functionalities and the sensory analysis. So as the majority, and I hope all of you know, basically a sourdough is made of flour and water. Historically, the majority of bakers, after the identification of baker's yeast by Louis Pasteur more than 200 years ago, gradually shifted to the use of a baker yeast instead of sourdough, which resulted in a loss of basically the heritage and the diversity associated to sourdough. Therefore, sourdough is nowadays used mainly, but not only, as a booster of taste in bakery products. And therefore, omics techniques are especially interesting to measure molecules that contribute to these functionalities and taste. So a sourdough is not a finished product. Basically, the sourdough goes into an oven with the dough, and through Maillard reaction generates new types of flavor and aromas that can be monitored with omics techniques. Then the focus of our research at Furatos is to basically make the link between these flavor precursors and the reactions that happen in order to better understand the molecular basis which is behind the sensory wheels that you see here on in this presentation, which are associated to each of our products. So here, if you're interested about the story of that product, I highly encourage you to scan the QR code and watch the video later. But as I mentioned, the introduction of bakery yeast has changed the bakery industry and the bakery sector more than 200 years ago. And one of the biggest challenge is to have a sourdough that gives the same leavening power as a yeast. And this sourdough, the leaven-up sourdough, is one of the strongest sourdoughs that exists actually on the market. It allows to fully replace industrial bakery yeast, which is Saccharomyces cerevisiae, and to use this sourdough through a 6-hour fermentation. Of course, the identification of this is was not based on serendipity. A whole part of screening of strains in order to identify the wild type strain that will provide this leavening power was necessary, and backed up by the sensory and flavor data analysis that I mentioned in the slide before. So another very important topic also related to the HealthFerm projects is how we can improve the nutrition of sourdough and bakery items. And basically, based on the previous presentations you have seen and the learnings that exist in the literature, what we do is that we identify what are the key changes that contribute to this nutritional improvement. So it is known that basically the time of fermentation boosts the production of healthy nutrients. The longer the fermentation, the healthier usually a sourdough become. And we can divide them in two categories. On one side, we have the release of free amino acids that occurs through the activity of enzyme, which improves digestibility. We have the gluten transformation that happens through, again, the activities of enzyme that has been shown by some studies to improve the digestibility. There are the prebiotic and the postbiotic potentials. There was a reference to it in the previous presentation. And therefore, the key molecules, for example, short-chain fatty acids, are again molecules that can be followed by omics technologies. On the other hand, there's also the glycemic index that can then be followed in individuals through clinical studies. And finally, there's the release of micronutrient, basically minerals, antioxidants that contribute to the improvement of the nutritional properties of a baked item. So probably the majority of you know that a grain is made of 3 parts, which are not evenly represented. And the majority of the refined flour is basically containing endosperm, which has a completely different nutritious profile than the bran, which is rich in fiber, and the germ, which is rich in minerals and vitamin. This has implication. So one can basically, by increasing the quantity of wheat bran, increase the fiber content and makes solutions that have a better nutritious profile as a product. However, we would like to focus on to bring that to the next level with fermentation. So here I'm showing the example of sprouted grains that are both sprouted and then fermented. The interesting part is that the unique fermentation that Puratos has is allowing to boost basically the nutritional profile by increasing the release of amino acids and the vitamin contents. So in this slide, basically we can see that some vitamins, so mainly B vitamins, not the majority of them, but some of them are increased following fermentation compared to the raw material, showing that the combined effect of sprouting and fermentation release some of them. And on the other hand, we can also see that the fermentation and the activity of proteases basically increase the amount of free amino acids that are present in the finished sprouted grain. Of course, this is what is present in the ingredient, and it is still interesting to focus on the implications of the baking on the stability of those metabolites. Another example of the use of omics techniques in the food industry is related to food safety and quality. And in this presentation, I'm not going to talk about the effect of mycotoxins, which are related to the fact that fermentation is not well controlled, or the fact that these molecules are mainly present in plant-based foods, but about the root cause analysis. So genomics is a very powerful technique to tackle that. And here I would like to take the example of an Italian startup called LabID, which is founded by Sparklis, the food tech venture fund that of Puratos, which basically leveraged the power of next generation sequencing to basically offer detailed and complete view of the microbial communities that are present in a sourdough. Based on these genomics techniques, they are able to provide biodiversity scores and compare the biodiversity of those sourdoughs, and they can serve as a quality control for private or professional bakers. Another example, and I will finish the presentation with two examples on food traceability and flavor and sensory analysis. So basically, one of the flagship initiatives of Puratos has been the sourdough library that is more than 10 years now, combined with the quest for the sourdough. So basically the sourdough library contains today more than 140 sourdoughs from around the world with the purpose of research and protecting heritage. Each of those sourdoughs are studied through omics techniques and characterized so that we maintain the knowledge for the owner and inspire further the community. And for the quest for sourdough, what I can say is that basically each sourdough, it's like wine. They represent the region where they're coming from, the terroir of flour, basically based on which they are made of, and they have specific flavor profile. And I will finish the presentation with a sweet note on chocolate. So as you know, chocolate is also a fermented product based on natural fermentation that undergoes special steps of processing before finishing as chocolate. And there are several opportunities to use omics technique. And the one I will focus on is related to the category of origins. So at Puratos, we have single origin chocolates that are made in specific regions. On this map, you can see 13 of them. They each are associated with a specific flavor and aroma profile. And one of the interesting studies that we do is to leverage the power of untargeted metabolomics to better understand what are the metabolites on the flavor precursors that generates and contributes those specific origins, taste, and flavor. So to conclude, the takeaway from the presentation is that there's a broad spectrum of opportunities and challenges in our sector that can be tackled through the variety of omics technique, and combining them, it's a very powerful way to generate new insights and allow this science-based decision making for innovation purposes. As you can see, these are complex and technical expertise that are needed. So trans and multidisciplinary research is needed, and a platform such as HealthFerm is key, especially in providing opportunities for open innovation and driving the food ecosystem forward. And I would like to thank you for your attention. We have open positions, so don't hesitate to check out our website if you're interested in R&D in industry.
Thank you very much, Azadeh, for this nice presentation, where you elaborate on what could be the role of all these techniques in the food arena. Maybe you can, you have made presenter. Okay, thank you very much. We have several questions to be addressed by all the participants, and one of those maybe rather easy questions to be addressed, Stefan, is for you. That is about can these genomic analysis also be applied to fungal strains, or maybe even broader to any microbial organism?
For sure, yes, that's a short answer. We do, and especially also within HealthFerm. We have done yeast research in the same trend as I presented, but that was on cocoa fermentation. So we do have also expertise in genomics on yeasts, but not sourdough strains yet. That's the part in HealthFerm.
Yeah, okay. Yeah. Kati, you have shown quite some examples of how to use metabolomic technologies. One of the questions that was addressed: how do you control for quality in your analysis? So how specific and are your measurements, and how sure can you be of the output from your technologies?
Yeah, I wonder whether the question relates to the study setup, the material used, or then the data analysis itself, but I guess it's the data analysis.
It's about LCMS outputs.
Yeah. So as I very briefly run through the whole pipeline, one essential aspect there is a pre-processing tool called Notame. So that is a tool that we have developed throughout the years, and that tool is particularly for the purpose of the data processing and quality control. So when we run the samples in an LCMS setup, something like maximum 300 we typically put on one batch. We always have the quality control samples, and quality control samples are the pool of a small aliquot of all the analytical samples. So these are collected and mixed as one sample. So that one sample then has a similar matrix than all the different analytical samples. And this quality control sample is first injected in the system as a priming sample for the colon and the system conditions, something like 12 injections. And after that, when we have all the different analytical samples injected, then something like every 12th sample will be a QC sample. Then after the week, when all the sequence has ended and all the data is collected, we collect the data from these quality control samples together with the analytical samples, and before going to any of the statistics or any downstream identification procedures, we utilize the signal collected from the quality control samples to stabilize the general level of the signal from the instrument, because these are long runs and that takes several days. It can be that the signal threshold in the system has variability during the run, and for that reason we always link it with the QC samples. And also we use the QC samples from a point of view that if we see something that is constantly always behaving in a strange way, it is only present in the QC samples or something like this. So whatever very awkward metabolite signals, we will then clean them out from the data matrix before going for the downstream statistics and identification. I hope this is at least this is one part of quality control, and yeah, I hope that covers what was asked.
I assume that the quality control issues with metabolomics, but also with genomics, is rather complex because of the complexity of the measurements that you do, of course. So if people want to have more details about it, I think they better contact you in person to get more details of these quality control mechanisms. I assume. With respect to another question, there is also to Kati. In rice sourdough, you showed there's a lot of peptides present in rice sourdough, and the question is, do you know if these peptides come from protein degradation or from microbial production? So can you identify actually by your system where these peptides are coming from?
Yeah, that's a very good question, and actually that was the first time for us to see such a huge amount of peptides actually in any sample. So it was a big surprise also for us, and since it's only very short peptides that you can detect with metabolomics technique, because larger polypeptides and proteins we precipitate them away. So only those that have been already spliced to quite many pieces are then there present. And our assumption is that yes, they are from the microbial metabolism, but the microbial metabolism that is using the proteins from the flour material. So that our assumption is that these are of the rye origin, but they are then produced by the microbes when they are using the substrates from the plant material. But which proteins do they precisely come from and so forth, that we haven't addressed any further. But surely these are very interesting because many of these dipeptides they can be biologically active and they might have a role. And I can say that later on in our animal feeding trials, we have seen that actually many of the most interesting compounds, for example, accumulating in the brain tissue of pigs after feeding sourdough rye bread, they are related to peptides and amino acids. So they do have a role, biological role, I would assume, but what precise, that's not yet clear.
Yeah, I can imagine that's different to identify, of course, what precise biological role these peptides may have, especially if you consume them, and if it has it to do with healthiness, has it to do with taste, texture, or whatever. And that's so there is also a question to Azadeh. You mentioned using sourdough as an ingredient for flavor instead of leavening agents. How much of the potential health promoting compounds will be present in such a process? Do you have any idea of or is that just a black box?
No, that's the purpose of actually doing all this research. So it will be to define how much, because we are a B2B, so we are not producing the finished product. So one of the main challenges would be to first identify what are the required amount that will be realistic without impacting taste and flavor in the finished product, which let's assume is bread, and then trying to see how basically the process, the bread making process, will impact that. So what will be the initial values we need to have in a sourdough so that we can have basically an ingredient on the market that could be associated to potential healthy benefits. So but that's the purpose of the whole project and many of the studies.
Stefan, maybe you can elaborate on what could be the role of the techniques that you use, the genomics techniques, on identification of potential health promoting or taste texture promoting compounds from these microorganisms.
The first key is to know the pathways and the genes that are contributing to those traits, and then once you have them, like I showed in the esterase case, once you have the genome sequence or the gene sequence and the amino acid sequence of the proteins of well-defined enzymes and well-defined genes, so all relates to the quality of annotation. And ideally, you don't have, you not only have the genomes, the sequences at hand, but you have also information from wet lab experiments where they characterize the enzymes. Once you have such sequences, well, then you can do a kind of genome mining and you can explore other genomes, other strains. But the starting point is to have well annotated, characterized genes and amino acid sequences.
Okay, just a very practical question popped up at the end of the questions row. Is are there also possibilities to use other grains? In this case, it was millet grain, but are there in the sourdough production? I assume that's possible. Eh? I'm asking Azadeh, maybe you can answer the question because you have a lot of experience.
Yes, it is possible to, I mean, I just showed two different sourdoughs from our range. There are definitely more, but I focus for the sake of this presentation, one which was focused on functionality and the other one which was focused on basically fiber content. But definitely you can make different mixtures with the sourdough, with grains, and you are not even limited to crops. You can go into legumes. So if you look, if you check the website of the Quest for Sourdough and the sourdough libraries, there are even sourdoughs which are based on chickpeas or different types of beans.
Yeah, so there are multiple options in that sense, and try to make our products more healthier, more tasteful, I assume. Yeah. Christophe, do you have anything to add to what has been said today?
No, I think we had a very good webinar, so I'm very happy with the contributions, and I learned a lot today myself, so that's good.
Okay, then I would like to close the webinar for today. Thank you very much for all the presentations, for the very good presentations, for the very good questions that have been posed in the question box. And once again, the webinar is recorded, so you can find it back on the website of the HealthFerm project if you want to look it back once again. Thank you very much for participation, and thank you very much for all the very nice presentation. And have a good day.
Thank you, Jan. Thank you, everybody.
Thank you very much.
Thank you. Bye-bye.
Goodbye.
Thank you. Bye.